Right now, as you listen to this, your brain is quietly losing something it cannot get back on its own. Every single day after 60, the cells responsible for building new neurons are shutting down. Not dying, not disappearing, shutting down, going silent. And when they go silent, your ability to form new memories, to learn new things, to keep your mind sharp, it all starts slipping away. Most people assume this is just aging, just how it works. But what if that shutdown is not permanent? Because in January 2026, a team of researchers found a single protein that, when restored, made aging brain cells start regenerating again. And what they discovered about how it works could change everything we thought we knew about the aging brain. I'm Dr. Richard James. I've spent more than 3 decades helping patients over 60 protect their most valuable organ, their brain. And what I'm about to walk you through today is one of the most promising discoveries I've come across in years. If you've been noticing more forgetfulness, more brain fog, more of those moments where a word is right on the tip of your tongue and it just won't come, I need you to understand something. That is not just aging. That is a biological process, a measurable one. And for the first time, we may have found the switch that controls it. This is not a supplement ad. This is peer-reviewed research published in Science Advances, one of the most respected scientific journals in the world. The team behind it works at the Yong Lu Lin School of Medicine at the National University of Singapore. What they found has stopped neuroscientists in their tracks. The average adult over 60 has already lost most of their neural stem cell activity. The cells are still there, they have just gone dormant. And every year they stay dormant, the gap between the brain you have and the brain you could have gets wider. But the real breakthrough was not just that they found this protein. It was what happened when they looked at how it actually works inside your cells. Because the mechanism revealed something nobody expected. Something that may open the door to treatments that simply weren't possible before. Stay with me through the next 17 minutes, because by the end, you'll know exactly what this protein is, how it works, and what you can do, starting tonight, to give your brain the best possible chance. Let's start with the problem, the one almost nobody talks about. Your brain is not a fixed machine. It was never supposed to be. Throughout your entire life, your brain has been building new neurons to support learning, memory, and cognitive flexibility. The cells responsible for this are called neural stem cells. They live in a region called the hippocampus, your brain's memory center. Think of neural stem cells as your brain's construction crew. Every time you learn a new name, store a new memory, or master a new skill, they are in the background, building the physical cells that make those experiences possible. For most of your life, they never stop working. But after 60, something changes. Those stem cells start going quiet. They stop dividing. They enter a dormant state. And when that happens, the supply of new neurons slows to a trickle. This is not a theory. This has been measured. Study after study in journals like Cell Stem Cell and Nature Neuroscience has confirmed it. The decline in neural stem cell activity is one of the central drivers of cognitive aging. The memory lapses, the slower processing, the brain fog. These are not random. They trace back to a specific biological process happening in a specific part of your brain. So why does it happen? Why do these cells, which have worked faithfully for 60, 70, 80 years, suddenly start shutting down? The answer has to do with something called telomeres. Think of telomeres as the plastic tips on your shoelaces. They sit at the ends of your chromosomes, protecting the genetic material inside. Every time one of your cells divides, those tips get a little shorter. After decades of divisions, they get so short that the cell receives a signal, a hard biological stop. And in your brain, that signal tells your neural stem cells to shut down. They don't die, they just stop working. Picture a factory where the machines are still on the floor, still plugged in, but the power has been cut. That's what's happening inside the memory center of your brain right now as you listen to this. And here's the part I want you to really sit with. This decline is what you're feeling. Every time you walk into a room and forget why you went in there. Every time a name slips away. Every time you read a page twice because nothing stuck the first time. That is the mechanism. That is what dormant neural stem cells feel like from the inside. For decades, doctors told patients this was just aging, inevitable, untouchable. But the cells are still there. They are just asleep. So the question becomes the most important question in brain health today. If the cells are still there, just dormant, is there anything that can wake them up? That is exactly what a team in Singapore set out to answer. And what they found was a protein that nobody had ever connected to the brain before. In January 2026, researchers at the National University of Singapore published something in Science Advances that stopped me in my tracks. Led by Assistant Professor Derek Ong and Dr. Yajing Liang at NUS Medicine, the team had been studying why aging neural stem cells lose the ability to regenerate, and they found a single protein that changes everything. It's called DMTF1. The full name is cyclin D-binding MIB-like transcription factor 1. Don't worry about memorizing that. Just remember what it does, because what it does is remarkable. DMTF1 is what I would call a regeneration switch. In young, healthy brains, DMTF1 is abundant. It keeps neural stem cells active, dividing, producing new neurons exactly the way they are supposed to. But in aging brains, especially in brains where telomeres have shortened, DMTF1 levels drop dramatically. And when DMTF1 drops, the stem cells go dormant. That is the link researchers had been missing for years. The missing piece between telomere shortening and cognitive decline. DMTF1 was sitting right in the middle of it the whole time, and nobody had seen it. Here's the part that made this study extraordinary. The researchers didn't just identify the protein, they tested what would happen if they restored it. They took aged, dormant neural stem cells, including human neural stem cells, not just mouse cells, and they artificially boosted DMTF1 levels. Then the cells started dividing again. Let me say that slowly. Dormant cells, aged cells, cells that had effectively retired. They woke up. They regained their ability to regenerate. The shutdown was not permanent. This was published in one of the world's most rigorous, peer-reviewed journals. The team used genome binding analyses, transcriptome mapping, and controlled aging models to confirm the result. This is not a headline from a blog. This is hard data from a top-tier laboratory. Now, I know what some of you are thinking. Another protein study? How is this different from everything else? And that is a fair question. I've seen dozens of so-called breakthroughs come and go over the years. Most don't survive contact with reality. So let me show you exactly what makes DMTF1 different, because the mechanism they uncovered changes the entire picture. And before you get too excited or too skeptical, you need to understand exactly how this protein works inside your cells, because the mechanism is what makes this truly remarkable. It's also what tells us something important about what you can do right now. Here is where the science takes a surprising turn. DMTF1 does not work alone. It activates two helper genes, Arid2 and Ss18. And these helper genes do something almost no one outside of molecular biology talks about. They physically loosen the packaging around your DNA. Let me explain what that means. Your DNA contains every instruction your cells need to grow, divide, and renew themselves. But in aging cells, that DNA gets locked down, tightly coiled, wrapped so firmly around its support proteins that nothing can read it. The growth instructions are still written there. Your cells just can't access them anymore. Think of your DNA like a book that has been locked shut. DMTF1 is the key that opens the lock. But DMTF1 doesn't read the book itself. It activates two helpers, Arid2 and Ss18, that physically open the pages so the growth instructions inside can finally be read. Without DMTF1, the book stays shut. The instructions are still there. The cell just has no way to get to them. This is part of what scientists call the SWI-SNF complex, a molecular machine that unwinds tightly packed DNA. When DMTF1 goes up, that machine starts running again. The DNA opens, growth genes switch on, the cell cycle restarts, and the stem cell, after years of silence, wakes up. But here is the finding that genuinely stunned me. Watch this closely. Even though shortened telomeres are what caused DMTF1 to decline in the first place, restoring DMTF1 did not require fixing the telomeres. The protein found a workaround. It bypassed one of the hardest problems in aging biology entirely. Think about that for a second. For 30 years, scientists have been chasing telomere lengthening as the holy grail of anti-aging research. It is one of the most stubborn, most expensive, most dangerous problems in biology. And this study just said, you don't need to solve it. You can route around it. That is a massive shift in how we think about brain aging. Now, I want to be transparent with you. This research is still in its early stages. These results come from lab-grown human stem cells and animal models, not from human clinical trials yet. The researchers themselves described their work as in its infancy. You cannot buy DMTF1 at the pharmacy. Not today, maybe not for years. But here is why this still matters to you right now. Because once you understand the mechanism, you can start supporting the same pathways through strategies that science has already validated. So now you understand the science. You understand what DMTF1 does and how it does it. But here is the question I know you've been waiting for. Is there anything you can do right now, today, to protect or support this process in your own brain? The answer is yes, and it starts tonight. Now, I know some of you are already halfway to the comment section, ready to write, Dr. James, this is a lab study. You can't buy this. How does this help me? And you're right to push back. I would be suspicious too. Let me give you 3 things to think about. First, the researchers were clear. This is early-stage research, in vitro and animal models. Human trials will take years. I'm not going to sell you a pill that doesn't exist yet. Second, DMTF1 is not an isolated finding floating in the dark. Other stem cell rejuvenation research, including work on Klotho, on plasma factors, on TET2, has consistently confirmed the same thing from different angles. Neurogenesis in the aging brain can be enhanced. The question is no longer whether brain aging can be slowed. The question is how. DMTF1 is one more piece of a puzzle that dozens of labs around the world are assembling. Third, and this is the part most people miss, the value of this study isn't a pill, it's a roadmap. Because once you know what drives neural stem cell dormancy, you can take evidence-backed steps to support the same pathways DMTF1 acts on. Steps that don't require waiting for a prescription. Steps that work through the same biological doors. Picture a woman, let's call her Dorothy, 68 years old, retired elementary school teacher, widowed, 2 adult children she doesn't see as often as she'd like. She comes to clinic convinced she's in the early stages of dementia because she forgot her grandson's birthday and misplaced her keys 3 times in 1 week. Her eyes are wet. She is terrified. This is the kind of woman I see every week. And what I want to tell her, what I want to tell you, is that the story your brain is telling you right now might not be the final chapter. So here is exactly what I want you to do. This is not vague lifestyle advice. This is a protocol built on the same science we just walked through, targeting the same pathways that DMTF1 acts on. Step 1, and this is the most important one, 30 minutes of aerobic exercise 5 days a week. Walking counts, swimming counts, dancing counts. What matters is that your heart rate goes up and stays up, because aerobic exercise is the single most powerful stimulator of adult neurogenesis that science has ever documented. It boosts a molecule called BDNF, brain-derived neurotrophic factor, which has been shown in multiple studies to directly increase neural stem cell activity in the hippocampus. If you take one thing from this video, this is it. This is not optional. Step 2, caloric moderation. And I don't mean starving yourself. I mean not eating to the point of discomfort at every meal. Research going back decades has shown that chronic caloric excess increases neuroinflammation and suppresses stem cell activity. If your doctor agrees it's safe for you, a simple overnight fast of 12 to 14 hours between dinner and breakfast can shift your biology in the right direction. Your brain responds to how much and how often you eat. Step 3, anti-inflammatory nutrition. Chronic brain inflammation, driven by activated immune cells in the brain, directly suppresses neural stem cell activation. An anti-inflammatory diet helps reduce that suppression. What that looks like in practice: fatty fish like salmon or sardines twice a week, daily berries, leafy greens, olive oil, and turmeric paired with black pepper to boost absorption. Cut processed foods, cut excess sugar. Your neurons will thank you. Step 4, quality sleep. 7 to 8 hours on a consistent schedule. Sleep is when your brain clears metabolic waste and consolidates new neural connections. Disrupted sleep accelerates neuroinflammation and suppresses stem cell activity. Set a bedtime. Protect it. Your brain is doing its most important work while you sleep. Step 5. Cognitive challenge. Learn something genuinely new, not crossword puzzles. Those are too routine to trigger real growth. I mean unfamiliar, effortful learning. A new language, a musical instrument, a new route through your neighborhood. Novel learning stimulates the hippocampus and promotes the survival of newly born neurons. The harder it feels, the more your brain is building. You are not powerless. Your brain is not broken. The cells are still there, and everything you just learned tells you exactly how to support them. I told you at the start of this video that your brain is quietly losing something every single day. That is still true. But now you know what it's losing, you know why, and you know exactly what to do about it, starting tonight. Your brain has been trying to tell you something for a long time. The forgetfulness, the fog, the moments that used to come easily and now take a little longer. Those were not failures, they were signals. And now you know what they mean and what to do about them. If this video helped you understand something nobody had explained to you before, do me a favor. Leave a comment and tell me which step from the protocol you are starting with tonight. Just one. That's how real change begins. And subscribe to the channel, because next week I'm covering another protein discovery, just released, that targets a completely different mechanism of brain aging. One that works alongside everything you just learned. You're going to want to see that one. I'm Dr. Richard James. Take care of your brain tonight. You've earned it. I'll see you in the next one.